Multiple analytical procedures were used at the end of this study for both pre- and post-slaughter samples. Serum samples analyzed at the Department of Animal Science at New Mexico State University. These were analyzed for cortisol concentrations, creatine kinase levels, plasma glucose levels, and plasma lactate levels. Furthermore, we also wanted to analyze the pH of our meat samples. A healthy pH is around 5.5 in normal meat. To do this, we removed a piece of the longissimus muscle that was closest to the anterior end of the strip loin. After 3 days post-mortem, 3 grams of that meat was sliced, and then deionized water was added until it was properly homogenized, and then the pH test was conducted. A series of statistical methods were employed in order to compute the correct results from all 3 categories that we were looking at. The 3 categories were behavioral scores, hormone concentrations or physiological variables, and muscle quality or meat quality. For all of these, we used the Remmel-based mixed effects model. We used this model because in our subject group, we were dealing with cattle from multiple sires and sexes. The Remmel-based mixed effects model estimates components of variance in unbalanced data. When looking at the WBSF scores, we used an ANOVA model to compute these results. The ANOVA model compared the means of the meats, and it told us if they were significantly different for each of the stress levels. Later, we used the Kenward-Roger approximation, which was used to calculate the correlations between physiological variables and the transportation variables. This was conducted at a P of less than 0.05. On the day of the experiment, the following conditions were present. The weather was dry and mild. The temperature was between 7 and 26 degrees Celsius. The loading onto the trucks took between 4 to 6 minutes for each of the groups. And the travel time to from the feedlot to the slaughterhouse facility was between 64 to 90 minutes. First, we can discuss First, we'll look at the behavioral results and the scores. The behavioral score ranged from calm to flighty. When humans appeared in the pen, 39% of cattle were calm, while 61% were considered anxious. When cattle were subjected to physical handling, 30% were calm and 70% were anxious, so it increased with physical handling. Next, we were looking at the correlation between physiological stress indicators and behavioral stress indicators. We found a positive correlation between high stress scores and high heart rate, respiration rate, rectal temperature, serum cortisol level, and serum epinephrine levels. All of the correlation statistic, or R values, were above 0.29 for each of the physiological indicators. However, the post-transportation behavior score was not correlated with circulating glucose and cortisol levels. The R values, which is the correlation statistic, were below 0.01 for both the glucose and cortisol. On the other hand, the post-transportation score was positively correlated with lactate levels and creatine kinase levels. The R statistic was above 0.28 for both the lactate level and the creatine kinase. If we look at our results, this is just a general overview of all of our data. Up here we have the data, which follows the cortisol levels and the epinephrine levels. On the far right side, it shows the post-transportation behavior. You can see that at calm, restless, and nervous, the cortisol and epinephrine levels have very little variation. Therefore, they're not significantly different. We can also look at the longissimus muscle pH scores. In general, we want the pH score of beef to be between 5 and 5.5. In our data sheet, it shows that between calm and nervous behavioral scores, the pH did not vary greatly. They all remained between 3.8, 5.38, or 5.37. Cattle with significantly elevated stress responses produced tougher longissimus muscle cuts than calmer cattle. In general, all the cattle categorized in the nervous behavioral score had longissimus muscle cuts of 3.72 kilograms, whereas cattle categorized in the calm behavior score had longissimus muscle cuts of 3.49 kilograms, and that was significantly lower. Additionally, carcass pH indicated that there were no dark, firm, dry cuts there. Therefore, our meat was generally healthy without considering the effect of cortisol and/or other stress hormones in the meat. And now I'm going to pass it over to Creston so he can discuss the discussion.
Okay, so in this study, we showed that the correlation between stress of handling, transportation, slaughter, on the meat quality of the cattle measured with the longissimus muscle WBSF, which was described earlier, and the stress from the handling, transportation, and slaughter activates two pathways. One is the hypothalamic pituitary adrenal axis, the HPA, and the sympathoadrenal axis, the SA. These are both activated when the cows are put under stress. The HPA stimulates secretion of the hormone ACTH, or the adrenocorticotropic hormone. This is secreted from the anterior pituitary gland and causes the production of cortisol by the adrenal cortex. When cortisol is released into the blood, it stimulates plasma glucose levels that activate protein catabolism, or breakdown, in the skeletal muscles. In the study, they did not find this to be any correlation to the decrease in meat quality. The second pathway is the SA axis. When this is activated by acute stress, it releases two neurotransmitters, epinephrine and norepinephrine. They are released from the adrenal medulla oblongata. Epinephrine activates beta-adrenic receptors, and this causes an increase in plasma, hepatic, and muscle glucose. It also increases the lactate concentrations, heart rate, body temperature, and respiration. Depletion of the muscle glucose is also a known factor for decreased muscle quality, and suggests that increased pre-slaughter stress causes the activation of the SA and also reduces meat quality. In conclusion, this study shown that plasma epinephrine concentrations, which is caused by stress, can cause heart rate increase, rectal temperature increase, and plasma lactate concentration increase. And the plasma lactate increase is the cause associated with higher muscle pH and a higher longissimus muscle WBSF and lower meat quality. The study also shown that nervous cattle from non-physical human contact caused an increase in stress, and this caused lower quality meat compared to cows that were more relaxed with the same situation of the non-physical human contact. And the same was shown in the study with the handling and transportation of them. And the reference to the article is there.